Okay, let's jump right in. Every time you grab a bite to eat, your body actually kicks off this massive, high-stakes biological demolition project. Welcome to today's Explainer. We're going to dive straight into the incredible molecular magic that happens the absolute second you swallow your food and how your system completely dismantles everything you eat.
Now, you might be thinking, isn't chewing enough? Well, you can chew all you want, but if your cells can't actually absorb the nutrients at a microscopic level, all that eating is kind of useless, right? Mechanical digestion, the literal physical act of chewing, only gets us so far. It breaks food down into smaller chunks, sure, but those chunks are still way too big for your cells to actually use. We need a deeper, far more fundamental way to get those nutrients out of the digestive tract.
So that's exactly where chemical digestion enters the chat. Basically, it's the breakdown of massive, insoluble molecules into tiny, soluble ones that your body can actually absorb and utilize. To pull this off, your glands secrete highly specialized enzymes that literally turn solid food into a fluid solution of dissolved particles. And I mean unimaginably tiny particles here, small enough to pass right through the epithelial lining of your small intestine and straight into your bloodstream.
Here is our roadmap for today's Explainer. We'll start with the big digestion problem, move into breaking down carbohydrates, then proteins, then fats, and we'll wrap it all up with acids, bile, and pH.
Section 1. The big digestion problem.
Okay, think about the solid food you eat like a massive, complex Lego castle. Your body can't just absorb an entire, fully built castle directly into the bloodstream. It just doesn't work like that. We need enzymes to act as tiny hands, pulling this massive structure apart piece by piece into single, easily transportable Lego bricks, hopefully without stepping on any in the process. Because only when it's stripped down to those tiny, soluble pieces can your cells actually put them to work.
Section 2. Breaking down carbohydrates.
So, digesting a long-chain polysaccharide like starch, it's a pretty incredible two-stage journey. Step 1 actually happens in your mouth and the duodenum, which is the first part of your small intestine. There, salivary and pancreatic amylase break the starch down into a 12-carbon sugar molecule called maltose. But we aren't done yet. Maltose is still way too big. So step 2 kicks in further down in the ileum. An enzyme called maltase, which hangs out right in the epithelial cell membranes, smashes that maltose down into single glucose molecules. Boom! Finally, a small enough Lego brick to be absorbed.
Section 3. Breaking down proteins. Now, when it comes to proteins, we have to rely on a totally different group of enzymes known as proteases. Their entire job is to chemically digest those large, tough, insoluble proteins into their soluble subunits, which you probably know as amino acids. Just like with carbs, the absolute goal here is taking massive, complex structures and relentlessly stripping them down to their fundamental building blocks.
And the way protein is digested is essentially this beautifully orchestrated relay race. It takes 3 different enzymes working sequentially across the digestive tract. Step 1 kicks off in the stomach, where an enzyme called Pepsin chops long amino acid chains into smaller molecules called peptides. Then, the baton gets passed to step 2 in the duodenum, where another enzyme, Trypsin, takes over to create even more peptides. Finally, step 3 hits in the ileum. A third enzyme, peptidase, finishes the job, breaking those peptides down into individual, soluble amino acids, perfectly prepped to enter the bloodstream.
Section 4. Breaking down fats. Fats, on the other hand, require a totally different strategy. Your body deploys an enzyme called lipase to break down fat molecules, specifically triglycerides, into 2 different soluble products, fatty acids and glycerol. Now, unlike that long relay race we saw with carbs and proteins, lipase is produced in the pancreas and does absolutely all of its intense fat dismantling work right there, localized in the duodenum.
Section 5. Acids, bile, and pH. Let's take a look at the stomach for a second, because it's essentially a brutal acid bath. The hydrochloric acid in your gastric juice serves this fascinating dual purpose. First, it's a hardcore chemical defense system, instantly killing harmful organisms that might have hitched a ride on your food. But at the exact same time, it's creating this highly acidic environment that the enzyme pepsin absolutely desperately needs to survive and thrive. Without that extreme acidity, pepsin just won't work optimally.
But this creates a major environmental conflict. Think about it. You've got this highly acidic mixture leaving the stomach and dumping right into the duodenum. But the enzymes waiting in the duodenum, like trypsin, they demand fairly neutral or even slightly alkaline conditions. They literally cannot do their job in acid. So how does the body instantly solve this massive pH crisis? Well, to counter all that acidity, an alkaline mixture called bile is released from the gallbladder where it's kept in storage. Bile contains a specific compound called sodium hydrogen carbonate, and it acts immediately to neutralize those acidic gastric juices the second they arrive from the stomach. It's just a brilliant chemical balancing act, perfectly prepping the environment so the pancreatic enzymes in the duodenum can work at their absolute peak.
All right, let's lock in everything we've covered today. We've got amylase and maltase handling the carbs. We have that powerful trio of pepsin, trypsin, and peptidase taking care of proteins across the stomach, duodenum, and ileum. And, of course, lipase independently dismantling the triglycerides. This right here is the ultimate master blueprint of how your body reduces your complex meals into sheer molecular fuel.
And all of this brilliant chemical deconstruction sets the stage perfectly for our next big biological mystery. We've used enzymes, acids, and bile to successfully dismantle these massive molecules into single soluble building blocks. But think about this. Once everything is broken down into these tiny pieces floating around in the ileum, how exactly does the body harvest them? How do these microscopic pieces physically cross over from the digestive tract into the bloodstream to actually power your life? We finished the demolition, but the cleanup and transport is a whole other incredible story. Keep pondering that, and I'll catch you on the next explainer.